Hi, I'm Ashley Gard. I'm a biomedical engineer at Draper, actively involved in projects that make use of Draper's human organ systems technology. Joining me today are three engineers who are involved in the development of our PREDICT96 human organ system technology. Do the three of you mind introducing yourselves and talking a bit about your role on PREDICT96?
Well, my name is Timothy Petrie. I've been at Draper for about 3 years now, and my current role on P96 is the technical director of a program in which we're trying to incorporate a number of organ models onto the PREDICT96 platform.
I'm Joe Charest, program manager at Draper. I've been here about 11 years, and I lead all of the kind of organ-on-chip human organ systems work, including PREDICT96 development.
And I'm Brett Eisenberg. I'm one of the lead engineers on platform technology development for the HOS system technology.
What is the PREDICT96 system, and what are the components of said system?
The PREDICT96 system is one of our platforms of organ-on-chip where we can grow tissues in the lab and make sure that they have the same behavior or function that they would in the body. It consists of a pump system along with a disposable plate and a controller. In addition to that, the cells and reagents that are going to allow you to form those tissues in the system. What this turns into when you have a realistic piece of tissue that functions as if it's in the body, it gives you a model that you can screen drugs or therapies, and it's going to be predictive of what happens in a human. So it gives you a lot of data early on in the development pipeline. And it's really important because you want to take a lot of the risk and time and cost out of that development process so that you can get therapies and medicines to patients faster.
Who conceived of the PREDICT96 technology?
Well, it's really hard to pinpoint it down to one person. It was a collaborative effort like all things at Draper, so it's really a team that did this. But it kind of started when we were making an organ-on-chip technology one at a time, the way most organ-on-chip organizations do. And we were asking some biological questions, and we realized we weren't going to get to the answer fast enough unless we scaled it up. And so we said, let's scale this to 96 of these organ-on-chips into one platform, and really leverage the throughput to get to the answer. We were given 1 year, and we turned it from a single unit into the 96 on one platform.
Some of the technology that's come through to help support PREDICT96 was developed on a previous program in which we were focused on making these sort of multi-organ systems that would allow you to study interactions of various tissues. And it was great in that we were able to generate a lot of unique biology. We were able to publish some papers on it. But the problem is it was sort of very academic scale, and we weren't able to sort of collect data at the rates that a pharmaceutical company would need to generate adequate data to make decisions on drug candidates and things like that. So we took a lot of the good things about that process and sort of moved it up and scaled it up to give us the sort of the PREDICT96 platform.
Do you mind sharing a bit about the story behind the PREDICT96 pump controller system?
We had an initial system in place that was developed by engineers for engineers, and it was probably overly complicated and needed to be sort of streamlined for an end user who didn't need to be able to tweak all the bells and whistles that come along with the old system. I sort of took that on to sort of streamline it and make it more user-friendly, and in that process started noticing all sorts of other things where we thought we could make improvements and just generally make the overall experience better for the end user. And as a result, I sort of took on this side project that grew into something larger than I anticipated, but it was a lot of fun and a big learning experience to sort of develop this whole new technology.
So what excites the three of you about these projects? Like, what gets you really excited and motivated?
So I'll start. Knowing the field fairly well, what excites me about the platform itself and the project is that they hold the three pillars that can really unleash the potential of the in vitro organ model field. And that's, in my opinion, maybe Joe may not agree with me, but it's usability, throughput, and the tunability.
So normally I like to disagree with Tim, but in this case I totally agree with him.
No.
And I'm excited about all those fundamentals, and that's what's been kind of baked into the platform from the start. But now that we have that and we start to present it to various people, I'm excited about the traction we're getting with various customers and end users. And they're finding uses for it that we totally expected, but they're also finding uses that are unexpected to us. So they're saying, oh, we can answer a new biological question, you've got enough throughput, we can ask the question, we can get enough replicates, we'll get the data, we can answer that. And they're kind of pulling it towards them and coming up with ideas of how it's going to be used, which is wonderful. We want to deploy our system and have it answer great questions and help, develop new therapies faster for people.
Do you all have a vision, and it's sounding like yes, for the future of PREDICT96, and what is that vision?
Well, that's what he gets paid to do. So my personal high-level vision, it's going to differ from other people, is I was saying, to expand the user base. So I want as many academics, biotechnology companies, pharmaceutical companies as possible to use it, also for our benefit, but also because the user feedback we'll get from that will allow us to improve upon that version. And one of the things that I've learned in my 3 years at Draper that we're outstanding at is to be able to revise our products very, very quickly. Where, as I said, that ideation to prototype period is extremely small. And so that the development cycle that we have is so short. We can incorporate customer feedback and customize that platform to those specifications. So I want to get as many hands as possible for that.
And then beyond that, the system is very adaptable, as Brett said, so we can start to incorporate new ways of collecting data so that we can measure things that weren't measured before, or measure them faster, or measure them in more real-time format, so that you can start to ask some biological questions that have not been answered yet. So I'm also excited about the technology creating a new area of study.
We have a core technology now that we're quite happy with. It's very adaptable, and there's a whole list of things that we would love to incorporate into the system, and it's new, whether it's imaging or sensing modalities and things like that. We'd love to keep just adding and adding and adding, and so that's what I'm excited to work on going forward.